Thank you so much for the invitation to come present to this, and I want to echo the previous speaker as far as the tremendous support that has been given by people in this room in helping us to be able to study area that we have found incredibly intriguing, but not necessarily something that had been the focus of a primary support organization like the NIH or the American Heart Association, whom I think are still quite behind when it comes to recognizing these issues about the specific biology in the different sexes. So I, at the sort of behest of Marcia, they gave a somewhat provocative title here about the question of whether estrogen and in fact also just sex hormones in general are actually a supportive and beneficial agent in this particular disease context that I will introduce and get you extremely familiar with, called viral myocarditis. So in a nutshell, viral myocarditis is a viral infection, much like what you think of as when people get a flu, except that in this particular case, the viruses actually go through the blood system and attack the heart in particular, and also they crosses the blood barrier. Here you can see what the blood vessel looks like, and through the blood vessels that come all the way into the tissue beds where your heart muscle cells are. In the very initial phases, these viral particles come into the heart muscle and they are taken up by receptors. So these are proteins that are found on the surface, and they bring it into the cell and causes immediate damage to the heart muscle cell. So that's the immediate acute phase. And then subsequent to that, as the cells are being damaged, they release lots of signals to tell the body, hey, we're under attack, please come and help us. These cytokines are being released, and the cytokine passes out through the circulation, which then subsequently brings in leukocytes with all the immune cells. And the immune cells become the next phase that actually come in and start trying to get rid of the virus, but at the same time not being good enough to recognize differences, so they also attack the heart cells and the muscle within the heart as well. So that second stage is you get another damage response from it, and then eventually leading to dilated heart failure. So here again is what I just mentioned. First phase, you have that immediate viral damages that are happening to the heart muscle that then is secondarily triggered by the immune responses from them, causes widespread damage in the blood vessels and also in the muscle, and then finally leading to dilated heart failure. And so there's been, over time, a variety of different viruses that have been studied, and I list some of these here that you can see. Some are actually quite common. So Coxsackie or enteroviruses, you probably have heard in the news in different contexts. In the same family, the Coxsackie also is responsible for hand, foot, and mouth disease, so somewhat less severe as the ones that attacks the heart muscles, but probably even more common adenoviruses. So these are viruses in your respiratory tract, and some of these viruses also have quite as dramatic of a damage within your heart as well. And here the rest are some of the viruses that have been identified to be associated with these viral muscle damage within the heart. And it's been a real conundrum for us in trying to understand, why is it that some people actually get this kind of severe damage, both early on as well as subsequently, as opposed to, the rest of the people who in general just gets a flu, viral-like illness, and then they completely recover and do perfectly fine. So it's one of the issue that I think we have been trying to address by both studying them in animal model as well as also in patients. So as Phil has shown you a little earlier about what these hearts look like when they're in the normal case, which is what you see on the left, normal size and function. This is one of the images in the echo for how the heart squeeze. The simplest, don't worry about all the anatomy aspect of this, is basically the ones on the left clearly squeezes much more vigorously where the muscle wall comes together, whereas the one on the right squeezes a lot weaker. So in about 20% of the patients develop this particular form called dilated cardiomyopathy from viral infection within 3 years window. And it's one of the most common causes for death among young people. So here, 8% of the cases in people less than 35 years old actually die, from their viral myocarditis. So, it's one of the problems that I think, we really need to find better ways to diagnose as well as treat patients. So at the tissue level, so here is a picture of what it looks like when you have heart muscle damage, both at the acute phases. Here you can see from the top section on the histology. So this is what happened when you take a heart muscle that has undergone these kind of damage initially, and you take a section and look at the actual muscle itself. What you see is there's quite a lot of inflammatory process here. So on the left-hand side, you see these damages from the immune cell that are infiltrating into the heart muscle, and the staining shows that where all those leukocytes, the immune cells, are. And then in the chronic phases, you get a lot of what's called scarring. So much like when you get damages in other tissue, you get scars, and in the heart you also get scars, and these scars are manifest in between the muscles, and there's some persistence of those inflammatory response in the heart muscles as well. And even in some cases, people have been able to identify specific viruses within the tissue itself that give the correlation between the fact that their heart damage is coming from the presence of these viruses that are continually ongoing, causing damage within the heart. So we would like to, trying to understand if there's a way you can actually see in the patient who have the diseases versus the one who don't, and whether they can be studied specifically for their responses with the virus. This slide is one of the data that supports how will people do, whether or not they actually have this inflammatory response. And I emphasize the inflammation aspect is that because while all of us have immune system, the ones that have ongoing immune damages are the ones that are have the worst outcome. So here what you can see is that when people do biopsy of the heart sections, the ones that are positive for this, the immunohistology or the inflammatory cell presence, have a worse survival, free from death or heart transplant, compared with people who don't have that persistence and presence of that immune response. And so with regards to the issue of the sex differences, so some of the epidemiological studies have been quite striking. So in this particular case, men have quite significant increase in frequency of myocarditis compared with women, and this starts from quite young. In this particular study from Finland, even in 15, 16 to 20-year-olds, it's sort of dramatically more in the men at that stage as women, and the numbers eventually evens out, and that might have to do with some of these differences in the immune responses. When they're younger, perhaps the immune system is much more robust and vigorous, and that contribute to the damage, whereas as you get older with age, those immune differences may eventually start to play a lesser role. But this impetus in looking at immune system is sort of what one of the questions that we have for research, which I'll study in the second. And then the other is whether the issue of the hormones, so sex hormone, may play a role in this beyond just the actual the sex, biological sex itself. So in animal studies, some of the early investigators have shown that if you take a mouse and treat the mice with testosterone, progesterone versus estradiol, there seems to be a significant increase in the viral presence and viral replication in the presence, of the testosterone, progesterone, as one indication that other than just the actual sex itself, there may be the hormone as being the contributor of that. And the other that's also been studied and found earlier is that men seems to have a more adverse profile of production of immune cells. So people have come to recognize that there are two different immune responses, one called the Th1 response, which are the active inflammatory aspect, and that are mediated by a protein factor that circulates in the body called interferon gamma, and men seems to produce greater level of interferon gamma compared with women. And then the females produces a different profile called IL-4, which is a different class called the Th2 responses, which actually are known to be more of a immune suppressive and modulatory. So again, some of the differences in protein profile that has already been noted. So for the research that we have that's been supported by the Women's Heart Health Program is we were hypothesizing that the sex hormones could play a role in regulating both the viral infectivity on the muscle cell from human sources, and also that there could be differences in immune modulation in response to those viral infection. And we wanted to be able to test that in this particular context of using viral infection. So we developed two goals for our study. So in the first aim of the studies is to look at that immediate acute phase where the viruses are attacking the muscle cells in the heart, and we want to look to see, are there differences in the way the viruses are able to get into the cell and multiply and make more copies of itself, and in the very first phase, and then subsequently in the second phase, when it's modulated by the immune cells, whether there are differences between men and women in terms of the immune cell response to viruses. So, as Phil had shown you a little earlier, we utilize the stem cell-derived muscle cell as a model. So in all of us now, we have the technology to be able to take a sample from you, like a blood cell, generate a new stem cell called the induced pluripotent stem cell that then can go through this differentiation process, involve multiple different chemicals and things added during this differentiation process, and eventually get the beating muscle cells that we can have plated into the dish. And as Phil has shown you earlier, we do this quite robustly, so we can directly study the heart muscles in the dish, which I think is quite a remarkable scientific advance that over the last 5 to 10 years we've now been able to achieve. And so one of the study from a graduate student that Joe Wu and I both mentor, Arun Sharma, has published a paper in looking at how to use these iPS cardiomyocytes to study the expansion, the growth, and also infection of the muscle cell as a way to model this particular disease of viral myocarditis. And in this case, with collaboration from Yan Carrot, who has developed a special version of this virus that actually reports a signal when the virus is present in the cell, so that we can actually measure the amount of the viruses that are actually in the cell. What we do is we take the virus and introduce into the heart muscle cell in the dish, and then we measure over time and see how much the viruses are in the cell and have expanded and grown, because these are live viruses, so they basically will grow on the backs of these muscle cells. So here you can see on the right-hand side is what happens when you do the measurement detection. So the redder the signal means the more the virus is, and this is an example when we do these different doses where we have greater amount going from the right to the left up to this point. The highest amount of virus, you get quite a robust signal for these viruses present at the earlier time point, and much brighter, as opposed to lower doses, you get much lower signal and at a much later time point after infection. So all represent that we can measure with an assay like this how much viruses are in the cells. So the first goal of what we were trying to look at is, does the sex hormones, so estrogen and testosterone, affect the expression of those receptors that are found on the surfaces of the muscle cells? Could that be one way that the hormones actually play a role in regulating the amount of infections that are happening? And then the second, if the viruses now have got into the cell, is there also a role the sex hormone plays in their ability to expand and multiply in the cell. And then finally, from the earlier study from Arun Sharma, we show that we are able to treat the viruses in these in the dish with antiviral drugs, and then see the responses of the cells to antiviral drugs. And we would like to study in this case whether the male and female cells respond differently to the antiviral drugs when infected with the virus. So those are the things we like to look at. So there's one very early preliminary data that we've collected so far, is that the receptor expression actually clearly reduces. So this represents a full decrease in the expression level when treated with estrogens. And what you can see is that, in fact, both male and female cells have significant reduction of the receptor for these viruses when treated with estrogen, and there may even be some differences in terms of more reduction in the female cells compared with male cells. So, again, I think these sort of preliminary data are quite encouraging. That tells us about some of the mechanism of how the female seems to do better with the viral infection. And then the second hypothesis for our project is looking at whether how these the sex hormones and affects the ability for the heart to respond. And so one of the early studies published have suggested that here you can see in a mouse study of the muscle damages in female are generally lower as a group compared with male, but specifically in the female when treated with a virus and estrogen, the effect is much less prominent compared with the female mice that are treated with testosterone, meaning testosterone may in fact be detrimental when in the presence of the viral infection, and generally females have lesser damage compared with males. So again, I think supports the epidemiology that seemed as the men having greater viral infection and susceptibility than female. And then with regards to the immune process, as I mentioned, the interferon Th1, which is greater in male compared with the IL-4, the immune suppressive in female, we're interested in finding out whether or not this Th1, Th2 responses are modulated with the sex hormones as well. So we develop a protocol to try to look at this specifically in patient cells. So our second aim to evaluate immune responses to collect patients who have previously had viral myocarditis and are now stable, but since they've been exposed to the virus, we know that their cells are now able to sense and respond. So we're collecting the blood from these patients. We're going to isolate the specific blood cells, the blood mononuclear cells, to try to then go through a special assay of profiling the different signals that are released from the cells, and then ask the question, are the immune responses that are seen and expected being similar between male and female humans, like what was seen in those earlier mouse study, and then also is there that prominent Th1 response as well. So a little bit about this process. So the assay is called Cytof, which was developed by Gary Nolan here. And the whole purpose of this assay is to be able to get extremely good precision on the expression of protein that are found within individual blood cells. So our study is going to look at taking the blood cell and split them into two different portions. One is just the blood cell as they are, and the other is the blood cell that have been now stimulated by treating with the viruses. And then we're going to do this profiling, the Cytof profiling, where different antibody that recognizes different proteins are uniquely tagged with a specific element, chemical, and then go through a mass cytometer, which basically will analyze all these different expression of different proteins to give us profiles of the different expression between male cells versus female cell, with and without the viral response, and then to look and see, are there the Th1, Th2 differences that I mentioned about that was seen in the patient cells who previously have been infected with the viruses. So a little bit of some of the progress we have so far. So we, as you see, successfully differentiated these cardiomyocytes from patients with healthy people as well as patients with viral myocarditis. We have shown that there's a reduction in the receptor expression when treated with estrogen. And we now also have 10 subjects identified who have this previous viral myocarditis to begin collecting blood sample to evaluate the response. So here are some of the demographics of the patients that we found. So you can see widespread of different ages from 20s into 70, 80s. All of them have reduced ejection fraction, which represents the heart function. And you can see that they're all decrease from normal, which should be in the 60s and 70s, and they all have a lymphocyte response from their immune system for the disease. So we're going to continue on and analyze and see what we find. So we are now in the process of completing the initial aim to look at these replication study with the imaging assay, as I mentioned, and also the effectiveness of the antivirals, which we're going through and evaluating now. We're also collecting and analyzing the blood sample from the patients to see their immune response. Down the road, we think in the future what could be really neat is to start looking at the heart functions with these cardiomyocytes. So we've been incorporating this aspect of the sex difference in some of the tissue engineering work, which is also ongoing within the lab. So we've been using innovative tools like 3D printing to be able to try to build tissues and as a way to model diseases and screen drugs. And we think that we can utilize that aspect of the platform but incorporate both male and female cells to try to look into whether there are differences in the contractile function and the responses with the virus. So, sort of really excited to be able to get the whole process and the project started and looking at some of these new exciting aspects of research. So, and certainly like what Phil says, all the works and certainly most of the work is done by people who are in the lab and also collaborator, and particularly Patricia Nguyen, who you'll hear from in a second about a different project that she also works on in sex difference biology. She's been a major contributor into the work, and this was in collaboration with Joe Wu's lab, Mark Davis, who is the one that has really utilized the Cytof technology, and Yang Carrot, who's been working on the Coxsackie viruses and has given us the reagent for that, Holden Macker for the Cytof, and as well as collaborator who has supplied us the patient to collect the samples from, and also members in the lab that help either assist with the assay or also with performing the assays. Again, really could not have done this without support from the Women Heart Health Program and as well as the Wisdom Centers generally with regards to sex difference biology and the other funding support from the lab to work on this area. And thank you so much for your attention.